So I will now introduce Dr. Armstead. Dr. Mindy Yeager-Armstead is a professor in the Department of Natural Resources and Environment at Marshall University in West Virginia. She's an aquatic toxicologist whose research primarily involves evaluating the effects of disturbance on aquatic ecosystems in the Appalachian region. Recently, her laboratory has focused on several types of disturbance, including the impacts of land development on benthic macroinvertebrate communities in headwater streams, non-traditional or emerging contaminants, and methods developed for sensitive biological endpoints. Currently, Dr. Armstead's research is focused on understanding algal blooms in riverine ecosystems with primary objectives of determining the role of tributaries in mainstream bloom formation and determining the conditions favoring bloom formation in wadable and non-wadable streams. Her undergrad degree in biology came from University of Charleston, West Virginia, and she obtained a Master of Science degree from Marshall and a PhD from Virginia Tech. Without further ado, I will then turn over the seminar platform to Dr. Armstead.
Thank you. And thank you all for having me. As I was saying before we got started, I'm just getting our master's program up and running here at Marshall with Natural Resources. So I'm anxious to share what we're doing in case anybody's interested in joining us. Okay, so I'm going to start out by talking a little bit about harmful algal blooms. I know everybody's heard of them, just so we all get on the same page. This is what I'm currently working on. And I'll tell you how I got started into this in a minute. But an algal bloom is a rapid growth of algae species that has a deleterious effect on the ecosystem or on users, which could be human users. The harmful algal blooms will cause restrictions for the uses, drinking water, recreation, ecological, and they do occur in all 50 states. And they occur in marine and freshwater environments. So some of the most famous ones you've heard about have been along the coast down in Florida in the last couple of years. The harmful algal blooms can degrade water quality in different ways. First of all, there's the consumption of oxygen. So algae in the water, they photosynthesize, they also metabolize. So during the day when they're photosynthesizing, they're creating oxygen. You often see supersaturated conditions. But at night, when there's no oxygen production, but oxygen demand, you can have oxygen sags that will create mortality in local aquatic organisms. Also, when a large algal bloom will die and they die off, the decomposition consumes oxygen, so you can have an oxygen sag. And that's one way that they can impact an environment. But a more kind of insidious way is that many of the cyanobacterial blooms will actually produce toxins. The toxins can be hepatotoxins, neurotoxins, they might just cause skin rashes, depends on the toxin, there's several different kinds. They are can be fatal to dogs or humans. And so this is a pretty significant threat that can happen in these blooms. Some of the, just a kind of a recent information on how many of these things we are actually dealing with. This is from 2017. The EPA reported there were 281 notices for freshwater HABs that included cautions, warning, public health advisories, and other health warnings. And so that's quite a lot of things being shut down or altered uses because of HABs. And this is just kind of a way that in our, in science, we can use to tell how much interest there is in a topic. And so you can see the number of publications from 2000 to 2020, how they've increased with respect to HABs. So this is something that is, ongoing and growing as an area of concern. A couple of high profile examples to show kind of the breadth of the problem. A lot of people heard about the 2014 bloom in Lake Erie that caused drinking water shutdown in the city of Toledo. So that left people without drinking water for 2 days or more and impacted of about approximately $65 million in lost benefits. And then in the summer of 2016, and again last summer, there were massive outbreaks in Lake Okeechobee, which is in Florida, and it drains the sugar industry, but it drains to both the Gulf Coast and the Atlantic. And so you had a real weird situation there. We have freshwater HABs happening in the lake. Then they go out into the ocean and they exist there briefly. And that then fed the marine HABs. So it's crazy right there. But then also in 2018, the city of Salem, Oregon, had to issue drinking water advisories because of toxins in their water. And you don't think of Oregon as being a big place where you're going to get these HABs. It's very widespread. And in 2015, it came to us in where I live, and you know, it impacted a lot of drinking water, where there was a 650-mile bloom of cyanobacteria with the toxins in the Ohio River. Okay, so this is a river bloom. A lot of what we just talked about and a lot of what you've heard about have been blooms in lakes and reservoirs. But in 2015, 650 miles in a flowing water system, this got everybody's attention because that's not supposed to happen. Now, I come from a wadable streams background. Most of my research has always been in the wadable streams. And we live and die by the note. That is our Bible, that we have, you know, the primary productivity in the headwaters is low, and you have allochthonous input from the leaf litter. In the mid reaches of the stream, you're going to get, that's where your primary productivity is. And in the lower reaches of the stream, you don't have productivity. There's very low primary productivity in the lower reaches of the stream. That's what I knew. And I was then became involved in this when the algal bloom came into our region. Then a lot of us, you know, became involved in it. And this is when I started to learn about large river systems. So I kind of reinvented a bit of myself by studying up on these large river systems and how Van Oort didn't really describe them. There's a lot of theories on productivity in large rivers and where it comes from. And many large river researchers feel that productivity is part of the riverine system, the larger river systems, which Van Oort didn't really include, but Thorpe and DeLong and others, they talked about how you can have autochthonous inputs in large river systems. You can have it in the backwaters, you can have it in the oxbows, you can have it along the banks. And so there were several discussions of the way that this productivity could occur in large river systems. The Junk et al. Said that, you know, when you have a flood or rain event, you have lateral input. Reynolds and Darcy suggested that in areas like in the internal, on the inner side of a bend where you have slower water, you could have some hydraulic storage. You could get phytoplankton there. And then the river ecosystem synthesis kind of tied all of the theories together to say where the productivity might come from in a large river system. But looking at these things and studying them, I didn't see anything that would support a 650-mile bloom in a river. Everything was, you get a little bit from here, you get a little bit from there, maybe you have something here, but nothing to say that this whole river is going to turn into an inoculum, a mesocosm for a 650-mile bloom, which, you know, it was very intense. It was very thick. It wasn't anything that was being described in these models. So I want to suggest here, and I haven't written it up yet, but you're hearing it here first, that these managed river ecosystems represent a significant difference from the river systems that we have described previously. They have bidirectional flow, and I'll describe that in a minute. They have unpredictable spatial and temporal flow patterns because of the locks and dam structures, which shut down the longitudinal movement of the water and create backwater conditions, almost lake-like conditions in parts of the pool. Because of the locks and dam structure, the pulse or the inflows from the tributaries, the oxbows, and those types of slack water areas, it's disconnected from flooding per se. It's connected to the lock and dam movement. So when they open the locks, the water draws in. And when they close, the water stops. Now, I saw this in my youth one day when I was working in Davis Creek of the Kanawha River. And I was standing there, and this was been, I don't know, the early 2000s. I was standing there trying to figure out what was wrong with my Marsh-McBurney flow meter. It's the workhorse of flow meters. There's no reason for it to have been giving me attitude that it was. And I'm trying to figure out what's wrong with this because I'm not going back without the data. And while I'm standing there, I see this beer bottle kind of go chugging right back upstream past me. And first I thought I was in the Twilight Zone and you know, my friends are playing a joke or whatever, but then I realized, no, the water's actually flowing the wrong way. I'm in the mouth of Davis Creek, 20 miles or more from the Winfield locks and dams. And the lock and dam structure had closed and the water was backed up to where it was moving up into the mouth of this tributary. That was the first time that I saw this happen, but I didn't really know the significance of it until, you know, recently. So there's a permanent alteration in that unidirectional condition of the river. And that affects the tributaries as well as the main stem and what's happening in the tributaries. And also in these run of the river systems where you have these locks and dams, you get three kind of different habitats in the main stem. You get the riverine system at the upper end of the pool, and then you get like a transitional zone, and the lower end of the pool is lake-like, more lake-like than it is river-like. And so we have three different habitats in the main stem, and then we have the influence on the tributaries that is really creating these conditions, and it's a permanent condition. So I'm interested ecologically in looking at what is happening in these systems. This has got to be different than what's happening in a flowing water system, right? Because in the flowing water system, we're talking about phytoplankton here. We're not talking about anything that's attached. So in a flowing water system, that phytoplankton is moving on down. You don't get a bloom from that. But in this system, how much is the algae in place? Can it develop a community? What's happening in these managed river systems? That's what I'm interested in from an ecological standpoint. Of course, I try to tie my research with applied questions so that you can get better funding for it. And that's kind of what I'm working on right now, is addressing this from a standpoint of what are we going to do about these harmful algal blooms? And then my personal interest in the ecology of the managed river system. The Ohio River is like an excellent place to study this. It is a series of locks and dams creating pools. So we have 20 different locks and dams creating pools. And my study area, can you guys see my mouse as a pointer?
Mindy, can you move it?
No?
No, we can't. No.
Oh, okay. I don't really know how that works. If you look in the center of the slide, you can see the green up locks and dams. And right in that pool, that's a 60-mile pool, which the pools are named by the lower lock. So that's the green up pool. And you can see in purple there, just in the center is the Huntington. Huntington is where Marshall's located. So this pool is right on our banks. And this is our main focus study area for our research. But you can see how the Ohio River is very much a managed river system. So I want to expand the riverine ecosystem synthesis model to better describe what's happening in managed rivers, including the hydrology, so that we can use that also to develop predictive capabilities and identify bloom conditions, possibly then developing management strategies for bloom avoidance or abatement. So that's kind of the driving factor of the research that I've been working on. And I want to point out my collaborators in the SENSE grant, which I'm going to be talking about. That grant was with University of Kentucky and Murray State University. And then in the deployment of my infrastructure, I collaborated with Orsenko, the Army Corps of Engineers, and the city of Huntington. And in the new research that we have funding for currently, which we're doing going forward, I'm collaborating with University of Kentucky, Auburn University, Mississippi State University, Tennessee Tech, and at Marshall's we have engineers, biology, and natural resources all working on this project. So First Sense, this was a grant that we had that went on for 5 years. It ended in 2021. And it was sensing and educating the nexus to sustain ecosystems, a partnership between West Virginia and Kentucky. It was an NSF Track 2 project where we were developing infrastructure in our regions. It promoted research collaboration. So we had 3 universities, 2 states. We worked with engineers and biologists, and we had sampling locations and monitoring sites in lakes, rivers, and streams. And we also did training for future researchers. We worked with early career faculty and had REU workshops. So it was a pretty big grant, went on for 5 years. And my part of that grant, my infrastructure deployment and my goals were focused in the Ohio River. The SENSE grant was a food, water, energy nexus project where we're looking at how do we manage the intersection of these needs for society with our resources. And the Ohio River is a perfect place for that because it has 38 power generating facilities. 25% of the watershed is agriculture, pasture, or row crop. There's 5 million people served by drinking water use, but 22 million people using the river for navigation, recreation, and drinking water. And also it has ecological uses. There's 160 species of fish, 120 species of mussels, some of them endangered, many of them threatened by invasives, zebra mussels and the carp that are moving through. So a lot going on in the Ohio River. It's an excellent kind of laboratory for us right on our doorstep. So the ecological goals or the research goals of the SENSE grant were to define the relationship between the land uses, agriculture, urban, and energy production in order to figure out those types of relationships so that we could maybe try to make things better, identify the environmental, physical, and biogeochemical factors that were creating water quality problems, and then leveraging that technology to better understand what was happening in the system. And I like this also because this grant required us to share our data, which I know a lot of people in science, they don't want to give stuff away until they're finished with it. So everybody kind of silos stuff, but this grant required us to make stuff publicly available. So we had our data broadcast. I mean, it was out there, anybody could take it and use it. I thought that was pretty great. And then ultimately we wanted to develop predictive models for the reservoir, the lock and dam system, and maybe try to work up some management scenarios where we could intersect with the harmful algal blooms. That's something we're still working on with our new grant. So in real time, we monitored with YSI exosondes, nitrate samplers. We had phosphate sensors. I'll talk about that in a minute. We logged the data, and then we had telemetry so it could go out onto websites and people could get it. We looked at pH, DO, conductivity, chlorophyll, and phycocyanin, which is a blue-green pigment, depth, photosynthetically active radiation, and nitrate. We got good data sets for those. We had hydrocycle phosphate sensors. We never did really get those things going to where we felt they were generating worthwhile data. So that was kind of a bust. Just put that in there so you guys consider before you buy them. The nitrate sensors worked great. We love those. So we generated a big data set with these tools. And in West Virginia, I deployed those on the lock and dam structures that bracketed the green-up pool. So on the bird locks on the upstream and the green-up blocks on the downstream, we mounted our equipment to the lock structures where we could access that to go out and download data and things like that and calibrate and that. So that went on for about 3 years, generating this giant data set. And so I can't really show you where this is, but if you look right at the where the white box says bird locks and dam, just in the corner of that white box, you'll see a tip of a bulkhead. That is where our equipment was deployed on the upstream bird locks. And similarly on the green-up blocks, if you look on the upstream just below the white box, you'll see again the bulkhead up there. And so our equipment was deployed up there. So I had to have our people had to walk out on those locks and try not to fall in. I hope they wore their life jackets every time, but I'm not 100% sure. But we were out there for quite a while, and we got a really good data set of what was going on in the Greenup pool. In addition to that, we did transect sampling locations within the pool to try to get a handle on the characteristics of the pool. And so we had 23 beginning at the upstream bird locks. We had 23 transects going to downstream to the Greenup locks. And we sampled these obviously under limited flow conditions because sometimes you can't get on the river. But we were trying to look at the critical conditions, which would be in late summer. And so we had several sampling dates where we would hit each of these transects. And at those sampling locations, we used the exosonde. We took a reading at the surface, half a meter below the surface, and then at 3-meter intervals to the bottom. We did algal and zooplankton grab samples, which we evaluated for chlorophyll analysis, counts, and IDs. And then we did total suspended solids grabs. We looked at the percent organic carbon in the suspended solids. We did alkalinity hardness, Secchi dish, and the algal torch, which is again a measurement, a fluorescent measurement of chlorophyll and phycocyanin. And then we recorded the physical channel characteristics. I'm going to present some of the data now, and I just want to tell you that I'm going to use some data from other sources as well. Discharge velocity and water quality from the USGS gauging station in Ironton. Sunrise sunset data from NOAA was used to calculate the gross primary productivity. Precipitation data also. And then Orsenko has been doing bi-monthly nutrient sampling for years at these locations, and so we also looked at that data. So here's a question: how productive is the Ohio River? Well, it just hosted a 650-mile bloom, so I was expecting that we were going to see some productivity. Here's the dissolved oxygen concentration. This is from the Greenup blocks. And you can see, well, what you expect, dissolved oxygen on the left panel. It is, related to temperature. So you can see in the winter it was higher, in the summer it was lower. That's what you expect. But I told you earlier that when you're talking about algae during the day, you expect supersaturation when you're having algal production during the day because it's producing oxygen. And so if you look at the right panel, that's the oxygen saturation at 2 p.m. in the afternoon, when you would expect to see really high oxygen values because algae was producing oxygen. And you don't see that. You don't, in the summer months, you don't even see, hold on, I think I'm on a timer here. You don't even see supersaturation in the summer months when it's hot. So that was the first clue that something's weird there. I thought we were going to have really a lot of algal production. So I looked at gross primary productivity. Nothing. Even in the summer months, those things, the box plots there on the left, they're bouncing around 0 for gross primary productivity. On the right is the ecosystem respiration, and these are calculated from the diurnal curves taken from the DO monitoring at the locks. And you can see that there's respiration going on. There are, especially in the summer months, you can see that at night, the respiration does drop down, and you can see that there's oxygen being consumed, but there's not really any production. And so finally, if you look at the chlorophyll analysis from the Greenup blocks, you can see that's around 2 or 4. Okay, the highest we ever got was one day we got a 36, not on this graph, but you see those highest dots in there, 10 micrograms per liter of chlorophyll. There's virtually no production going on in the Ohio River. And that's kind of like, what? I wasn't really expecting that since we just had this big algal bloom. So what is going on there? And I just wanted to put this in there to remind me that we also had phycocyanin. So we could see the presence of blue-green algae at very low concentrations, but also in the pool. So when you think about productivity and harmful algal blooms, you think about nutrient lights and temperature. Those are the factors that drive algal blooms, at least what we know of in lakes. But those things, we weren't seeing that kind of productivity. So we're trying to figure out now what happens, what's going on in this Greenup pool that we're not getting all that productivity that we expected from that bloom. And so I want to look here for a minute at what nutrient, lights, and temperature and how that might be impacting the algae. Well, these graphs show the Orsenko data set for the bi-monthly monitoring on nutrients. So the top graph on the left is nitrate plotted with discharge, and on the bottom, phosphate plotted with discharge. And there's no relationship there between the nutrients and discharge, which is, you know, kind of you wonder, well, we think those things are flowing in during high flow events and we don't see that. So that's one issue there that we now wonder, well, okay, where are those things coming from? We need to find an internal source or a consistent source. And then also, if you look at the relationship between nitrogen and phosphorus, it's pretty loose, not very significant. So that gives us some clue as to maybe where we start looking as sources. But if we look at nutrients overall, we see that they are not limiting at all in the system anywhere. And that is kind of a significant thing. I got to move your all's faces away so that I can describe the graph to you. So on this graph, we have nitrate on the y-axis and discharge on the x-axis. And you can see that there's not that much of a relationship between nitrogen and discharge, which we already just talked about. But the one thing that I want to point out is that when you look at the nitrogen, it's not limiting ever. There's no time when nitrogen would be limiting in the system. So there's plenty of nutrients for the algal community to grow. Temperature's on here also. And so you can see that there is a relationship between temperature and discharge. The highest discharges are generally during the winter, which is what we would expect. But nitrogen, not really being influenced that much by either. But the one thing to point out, nitrogen and phosphorus are never limiting. There's always a supply that those algae could be growing if other conditions were right. So looking at light, how does light influence the productivity in the Ohio River? So this graph shows the photosynthetically active radiation taken at depth in the transect sampling locations. And starting on the left, you have an August of 2018, July of 2019, August of 2019, and September of 2019. So we're going through the low flow conditions here, and we're representing the photosynthetically active radiation at each of the depths, the first bubble being the 0.5 meter, and then 1 meter, and then 3 meter intervals below that. So we're not getting good light penetration. And this is at transect 2, I believe it's behind your faces for me. But so this is in the upstream area where I told you that the pool is more river-like. Okay, so now I'm going to go downstream and show you some other transect light penetration graphs. I think transect 7, which is right where the city of Huntington begins, 13, which is towards the end of Huntington, and now that's in the transitional zone where we're going from more flowing water in the upper end to more pool-like conditions, and then the final transects will be towards the more pool-like conditions. And so here's transect 7 right up above Huntington, and you can see from the bubbles that you are getting light penetration to 0.5 meter for that first samples collected, and a little bit at 1 meter. Okay, and at transect 13, which is mid pool, then you are getting some light penetration, again, just in the upper meter or so. But at depth, you're not getting any light penetration at all. So very limited opportunity for growth for the algae this far in the pool. When you get further down in the pool, and the lower graph there is transect 23, which is right above the actual lock. And you see you're getting, again, very limited light penetration. So there's very limited area for the algae to grow and be exposed to the light that they need for photosynthesis. So obviously light is a limiting factor in the Ohio River pool. And that is something that we were able to define better from these samples. Now, temperature, this graph may be the first one that came out of this data set that actually made sense, where it's colder in the winter. It's warmer in the summer. Okay, we got that. That's great. That is what we expected finally. But then when you look at the chlorophyll, no relationship to the seasons. Sometimes you see a little algal bloom in the spring, and we do see that, but that's virtually it. We did not get any other, and this is a quarterly graph, but we have it by month as well. No relationship between the temperature and the algal growth. So the three main factors that we know usually influence algal blooms, nutrients, light, and temperature, don't seem to be that related, except possibly for the limitations by light. That does not explain why we got a 650-mile long bloom. So then we look at some other factors, velocity and turbidity. Obviously, turbidity is a problem because we saw those light numbers. But velocity and discharge in this managed river system. Obviously, an algae has to be able to reproduce faster than it washes out. Okay, that's good. So how does that play in this description? Well, when we look at flow, we can see that we obviously think it's going to limit productivity in flowing waters because, as I just said, it has to reproduce faster than it washes out. And when we look at gross primary productivity and respiration in these two graphs, this being the full data set and this being the low flow conditions, we see that under low flow, retention time is reduced. This slope of this line indicates a more lake-like condition under the low flow than the relationship between gross primary productivity and respiration under all flow conditions. So this shift towards a stronger relationship is an indication of more lake-like conditions are occurring at certain times of the year. And then we look at another one of these crazy graphs, which I'm going to try to move. Again, with this charge across the bottom and chlorophyll on the left y-axis. Okay, the color here is also chlorophyll, just to get a better visual image. And you can see virtually no real relationship between the discharge and the chlorophyll, although the highest chlorophyll values are occurring. You still get the at times when there's no chlorophyll, but the highest chlorophyll does occur under the lowest flow conditions. In rivers, sometimes that happens the opposite of that because the highest chlorophyll can be when things are washing in from the tributaries. So this is significant because it kind of indicates that the growth is occurring in the main stem. And so that's one potential contributor to conditions that might support a harmful algal bloom. But it's more complicated than that. When you think about the theories on riverine primary productivity, which put the primary productivity up into the tributaries, the oxbows, the slack water, and the backwater. When we have a situation where the locks are holding a high pool elevation in order to support navigation under low flow conditions of the tributaries, then that's what's creating the situation where the bottles wash back upstream. So the area for photosynthesis and phytoplankton community development is so much greater in those tributaries where this slack water condition is now being exacerbated by the elevation of the pool, creating additional habitat for phytoplankton development. So when we think about velocity and phytoplankton development, it goes farther than the riverine theories on slack water oxbows and banks. It's now moving up into the tributaries with the area of which being impacted is unknown. So a little bit of discussion of the turbidity. These are the messy data, so I like to put that in there. And you can see the chlorophyll and the phycocyanin, chlorophyll in the green and phycocyanin in the blue at the bottom there, put with turbidity on the second y-axis. They're all all over the place. The thing I want to point out is if you hone in on just the bottom right corner, which is low flow condition in August and September, what you're going to see is that the turbidity on the gray line there with the 0 on the right y-axis, the turbidity never goes to 0. 90% of the time, the turbidity is greater than 5 NTU, which is sufficient to limit photosynthesis, even under the low flow conditions. In this pool, the turbidity is never sitting at 0. Oh, sorry, I'm going the wrong way. So one of the reasons for that is the amount of barge traffic that's going through this pool. And I don't know if it shows up well on your screen, but this is a picture of a barge moving up the river. And behind the barge, you can see the silt plume as it is just churned up, as you know, this river is used for a lot of navigation. And so this may be contributing. I mean, large rivers always have a lot of turbidity, but this barge traffic might also be contributing, and we're not sure yet to what extent that may be. So if you look at this, and I promise this is the last one of these crazy graphs, turbidity on the left, velocity on the bottom, the x-axis, and chlorophyll is the color. And if you look in the lower left corner of the graph and you see the dots of color, there's not often that we're getting a lot of chlorophyll, but you can see that the highest chlorophyll events occur under the critical conditions of low turbidity and low flow. And so these are the conditions that we feel must have been necessary in 2015 when we don't have the data, because all of us got to this party later than that. But we can see the critical condition for the actual increase in chlorophyll is going to be under low turbidity, low flow. Now the question is, when does that exist and where does that exist in a managed river? So in the main stem, more likely that's going to be on the downstream end of the pool, but also in these tributaries. As the pool elevation is high in relation to the tributaries, then we are going to have slack water in the tributaries going upstream. How far? Not sure. Not sure the amount of area that will be available for algal growth under these conditions. So that's kind of where we are. At the end of the SINS grant, we've identified these conditions. We have data for these conditions, and now we are working, and we had been working to develop some modeling of that before, but in our current project with the additional collaborators, we just received funding from ERDIC, Army Corps of Engineers, in order to expand our evaluations and develop predictive models. So we have, we're using the data that we developed under SINS for hydrodynamic modeling. We are also looking at machine learning in two different ways. So we're using the large data set that we have to try some machine learning techniques and see if we can predict the presence of the favorable conditions. And we're also trying to use publicly available data to see if we can reach similar conclusions in predicting the presence of the favorable conditions. And the reason for that is because the Corps of Engineers is managing all these pools. I showed you all the pools in the Ohio River. Those pools exist in all the managed rivers because, well, we all live in the floodplains now. So most of our large rivers are managed. In West Virginia, all of us live in a floodplain. So most of our rivers are controlled in this way. And so for decision making, will the regulators or the managers need the level of data that we have developed under the SINS program or others, or can they make these types of management decisions and come to the conclusions of when these conditions exist using only publicly available data such as USGS gauging data and other publicly available stuff? So we're operating on that in two ways. And then we want to be able to develop these predictive models so that we can explore management scenarios, because the locks and dams offers the ability to control that flow, open it, move stuff out, keep stuff in. So can we operate management scenarios that's going to reduce our likelihood of developing a bloom? Or if we do have a bloom develop, can we manage it or mitigate it using management of the structures? And so that's kind of where we're going with the core right now. Now, for me, a lot of my research on the new grant is going to be in the tributaries. We're going to be using acoustic Doppler velocity surfboards. They're really cool, and establishing, what's going on in those tributaries. When do the tributaries become stagnant and slack, and when do they actually flow into the main stem? And what's happening with the phytoplankton in the tributaries? I mean, I've been doing research in tributaries for a long time, but we were not looking at phytoplankton. We were always looking at periphyton. And so now we're looking at a phytoplankton community that can be drawn in into the main stem when the velocity conditions change and seed perhaps the main stem blooms. And so we're beginning a monitoring and data collecting methods to try to establish what's happening in those tributaries. We're going to be looking at water quality, nutrients, phytoplankton. We have picked 4 tributaries where we're going to start getting this information. And then also I'm working with the microbiologist who's going to be doing some genetic stuff, but I'm really into the community ecology investigations. What species are present? How are they operating together? When is it shifting toward the green toward the blue-green from the green? And that's another thing I think I forgot to say on another slide, that we did identify conditions. We identified times in our monitoring. We didn't have other blooms, but when the blue-green algae rose in the community relative to the green algae. And so we would interpret that to say that's when the conditions are appropriate for the blue-green algae to bloom, when whatever was happening right then was favoring the blue-greens over the greens, and they were rising in relation. And so we're focusing on a few of those different areas to look at what was happening at that time. That's part of our modeling endeavor. So this is what we have going on. We are just getting started. This was funded by the Corps, like I said, 2 weeks ago. And so we're just staffing up, gearing up, and getting our methods in place and everything like that. So it's pretty exciting. And with that, I will answer any questions. That was kind of fast.
Thank you, Mindy. Are there any questions for our speaker folks?
Brady has one there.
So I'm just going to ask, I was curious about, as far as a lot of the presentation focused on the impacts on the phytoplankton, as far as the algal blooms. Just because I had been working in your lab, how are we seeing these blooms in recent years correlate with the abundance and diversity of, say, the macroinvertebrates, if you have that data?
Well, we weren't doing macroinvertebrates in the Ohio River.
Okay.
There have been studies on the toxicity of the cyanobacteria to the fish, and they're not as toxic to some of the other organisms as they are to humans. So the focus has really been more on human health.
Okay.
But I don't know that answer in the tributaries. Since I've been involved in the main stem and in the large river stuff, we haven't done much macroinvertebrates. You know, Tom Jones here at Marshall, he works on mussels, and he has several projects on mussels, but has not investigated what impact the bloom. There was only the one big bloom. There's been some smaller ones, but that was the main one. Haven't looked at the impact of that on mussels. To my knowledge, there's very little research on that. But okay. I mean, there's research on the periphyton with respect to bug preferences on what algae they'll eat, but all of that's occurring on rocks. So, I mean, it's not the phytoplankton in the larger rivers is kind of the anomaly. So, but yeah, okay. But we haven't done that. And we do benthic macroinvertebrate sampling in Four Pole Creek. So, and that's one of our sites for this. So we may develop some of that. But to my knowledge, no one has put the phytoplankton together with that.
Okay, I see. Thank you.
Mindy, I've got 2 questions. So these algal blooms occur on managed rivers, of course. We have all these locks and some still waters. In terms of natural flow rivers, are algal blooms ever a situation with those, since they have much higher flow through, I would imagine?
Well, in West Virginia, we have attached algal blooms. So you'll have a green algae bloom in a flowing river, but in the flowing water, free flowing particularly, a lot of times it's shallower water and you're getting the periphyton. So we do have periphyton blooms in high nutrient, flowing water. But in the large rivers where it's too deep for the periphyton community, no, you don't have that kind of attached thing.
Okay. And you mentioned trying to work, building predictive models regarding maybe increasing flow when you have a potential situation where you can get these algal blooms. A political question: how receptive do you think the stakeholders would be if you asked them to modify the water flow, given the navigation needs and things like that? Have any sense about how people might, in the public sector, might actually view your recommendations?
Yeah, we have talked with the Corps about that. And a couple of things. First of all, they are, they obviously their first concern is navigational pool and maintaining their objectives. And they have plans, objective, management strategies for all the locks with those objectives and goals. But recently, and so I'm going to say their position is that their first priority is navigation and maintaining the pool in the way that their plan describes.
Right.
But recently they have a sustainable rivers project where they are starting to look at how they can improve habitat in these rivers by managing flows for other things, either elevating the pool or dropping the pool. So they have, and the Ohio River is one of them, several rivers where they are actively looking at ways to maybe change the flow in order to facilitate ecological resources.
Oh.
They haven't come up, we haven't gotten to the point where we're like, oh, what about an algal bloom? Because we don't have those predictive things yet to say this could help an algal bloom or not. But hopefully from the results of our study, we may be able to make management strategy predictions of things that they might consider. And because of the Sustainable Rivers program, I think they would probably, consider that.
Okay, neat.
Not speaking for them though, but they've cracked the lid. Also on Lake Okeechobee, they did make alterations to their operational plan for the purpose of bloom avoidance. So it's out of the box.
That's great. That's great. That's highly encouraging, quite frankly.
Yeah.
I'm familiar. My brother lives in Florida, and I'm familiar with the runoff from the sugar plantations, and those algal blooms are getting down there. They're just devastating, especially in estuaries and the east coast of Florida. It's just been, as far as the impact on local economy, tourism and fishing and things like that.
I just was at a workshop with some ladies that worked in the Mississippi for the state. And they were just saying, how devastating it is to their state when they have to close beaches.
Yes.
I mean, yeah.
One final question, Mindy, if I may. Looking at, global climate change, increasing temperatures, do you have a sense from your work, or do you want to project what might be the consequence of increasing riverine temperatures over time as far as these algal blooms go?
Well, in theory, the algae should grow faster when it's warmer. I mean, that would be conventional. That's not what we saw right here, right now. But in theory, that would, things metabolize and grow faster when it's warmer. So all other things being equal and nutrients never being limiting, and they're not, then you could project that those things would get worse. Maybe in lakes where I haven't really worked in the lake habs, maybe they follow more of that convention. So far, we haven't seen that here, but we haven't really seen another bloom here. When I was talking to you about that, here's what what it showed with temperature, that was not much algae.
Okay.
So there's the theory of what we think should happen, and then there's what actually did happen. So
yes, the nature of science, of course.
Yeah.
Thank you, Mindy. Any further questions for our speaker?
Stop sharing my screen there.
Okay, seeing none, Mindy, I thank you for sharing your research with us today, most enjoyable. Folks, thank you for your time, and then we will resume our